please join me in welcoming Professor Robin May.
Wow, I get whoops. That's quite exciting. Good evening. Thank you very much for joining here and online, wherever you are. This is my sixth and final lecture in this series, where those of you who've been before, we've been rummaging around immunity in all its colours, human immunity, plant immunity. Last time we did computer immunity. And today is my kind of free license to speculate wildly, because today is about the future of immunity. And the good thing about the future is that it hasn't happened yet, so you can't tell me I'm wrong. Not yet, at least, anyway. And what I really want to do is think about what the future might hold for our immune systems and the kind of two extremes. So are we going to be living to 350, disease-free, rampantly healthy for our entire lives, or are we all going to die in a horrible pandemic? And those are your two extremes, and hopefully the answer is somewhere between the two. So we're going to think about the future of immunity. But sort of like, I don't know if people are into hill walking or mountain climbing or something. If you go hill walking, it's really important, I think, sometimes to stop and look back and admire the view, right? Otherwise, you spend your entire life thinking, God, this summit is never coming. And so like that, I think before we start thinking about the future of immunity, it's worth taking a couple of minutes to think about the past of immunity, and in particular to celebrate, I think, what has been just the most amazing set of achievements of science, largely, and human behaviour over the last couple of hundred years. And so I'm going to start with this graph, which is from the Office of National Statistics. And this is a slightly complicated graph, which shows you in colour coding the primary cause of death across age groups over the last 100 and bit years, back to 1915. And what you can see, I think, quite straightforwardly, without even reading the key, is that in 1915, green is infection. In 1915, at any age, almost, the thing that was most likely to kill you was an infection. When you were born, right through, unless you made it to the ripe old age of 75, which in 1915 was quite impressive, at which point you instead succumb to this slightly dubious phrase of senile decay. I'm not sure if I want an infection or senile decay, but either way, not great. But infections killed the vast majority of people at all ages in 1915, and actually for quite a long time. And then bit by bit, you see over time the green bars disappearing as we start to tackle infections. And quite remarkably, I think, by 2015, so 10 years ago, infections were not the leading cause of death for any age group in the United Kingdom. I think that's quite remarkable. So we've gone from killing almost everybody to killing a tiny minority within 100 years, which in evolutionary terms is a kind of a flash in the pan, right? And that is done primarily by three things, right? So part of this is about human behavior, in particular social factors. We got better at putting people into nice houses. We got better at things like public sanitation, so on and so forth. So partly it's that. Partly, of course, it was about antibiotics, which came in about here. And you can see this sudden drop between 1945 and 1955 in infection rates as we started to roll out penicillin initially and then subsequently other antibiotics. And lastly, but perhaps most importantly, and for those of you who came to my vaccination lecture, you will have heard me flag wave for vaccines already, but this is about vaccination. 1915, pretty much no one was vaccinated for anything really. Today, we're all vaccinated. When you have a baby today, typically you're vaccinated for 20 or 30 diseases by the time they're going to school. And vaccinations have transformed our risk of infection. And you can see that here with just two examples. So here we have diphtheria and measles. Back in 1915, if you were a typical GP in a typical town in England, you would see loads and loads of cases of both diphtheria and measles. And sadly, a reasonable number of your patients would die of it. Today, happily, if you're a GP, you might go many years and not see either of these cases, happily, and hopefully you'll never see someone die of them, although not entirely true. And you can see this huge drop-off here, which is both a result of vaccination. So particularly, I think, for diphtheria, within just a couple of years, we've gone from 70 or 80,000 cases down to essentially zero. So before we think about the future of immunity, I think we should stop and celebrate the fact that the past of immunity, the last 100 years, has been pretty spectacular. So the question is, can we keep doing this? Can we keep driving those kind of changes by harnessing immunity going forwards? And if we think about that, if we think about immune systems, these are biological systems, right? Your immune system, just like your heart or your lungs or your digestive system, it is a body system, it is a biological system, and it's shaped by the same evolutionary forces that shape all the rest of things about us. So broadly speaking, if we're thinking about the future of immune systems, there are only really two ways in which our immune systems can change. One is through natural evolutionary process, so evolution, and the other is through what I've loosely called innovation. So that is ourselves, humans, coming up with creative new ideas like antibiotics that might change the shape of immunity going forward. And so what I want to do tonight is think a little bit about both of these and think about how they might play out. So let's think first about evolution. Actually, a question I quite often get from people is, God, we've been around for a long time as humans. We've had colds and flu and all this kind of stuff for a long time. Why haven't we just evolved total resistance? Why do we still get colds after all these thousands of years? And the answer to that, of course, is because that's not how evolution works. Evolution works on this process of competition and this process of interaction between different living organisms, which all infectious diseases are. The other thing about evolution is that it works in a way that people often sort of fail to appreciate is kind of quite brutal. So here's my quick recap, one slide recap of evolution. And as you can see, I've employed my very best CGI graphics here. So this is my live organism. All living organisms are subject to evolution, and essentially they all are subject to it because of two things. The first thing is that all organisms generally over-reproduce. We all produce more, on average, more than one offspring each. And therefore, every generation, there are more new offspring than there are current adults, right? And which means it's competition. It means you have more offspring than the planet can sustain, and they're competing. And the second principle is that when you have offspring, they are never completely identical. Even if you're an organism like a bacteria that tends to divide by clonal propagation, so not through sex, but through just reproducing itself, you still don't produce identical offspring because of things like DNA mutation and genetic recombination. So all organisms produce lots and lots of young, and those young vary. And then, as you all know from school and from learning about Darwin, the principle here is that you have lots of offspring, more than can be sustained. They compete. The ones that happen to have inherited the best genes for the environment they find themselves in survive. The other ones die. The ones that survive have more offspring. Those offspring vary. And once again, the same process happens again and again and again. And over millions of years, what you end up with is this wonderful thing, which we call the tree of life. And that is the basic principle of evolution, right? There is competition and there is survival of the fittest. Now, if we think about that, in the context of immunity and how our immune systems might evolve, there are basically only two forces that are likely to change things in our immune system by evolutionary processes. And they are essentially sex and death, right? There are two ways by which a gene will spread, will become more abundant through evolution. One is if you have more successful offspring than average, so you're quite successful here. The other is if offspring are removed from the population through death. So if you have for example, a new immunity gene, the only way that gene will kind of spread through evolutionary processes is A, if it provides an advantage and protects you from dying, whereas everybody else dies more frequently, or B, if it makes you massively more likely to have lots of successful babies. In principle, this is theoretically possible. In practice, I'm struggling to think of an example of an immunity gene which makes you the most desirable thing to the opposite sex you can imagine. But I guess it's conceivably possible. If anyone has an example, I'd like to hear that. So unfortunately, most of what we're talking about tonight is this one over here. So immunity is about not dying, primarily. So if we think about, therefore, what might shape the immune system going forwards, what we're thinking about are genes that confer a survival advantage, and typically a survival advantage in the face of infection. So if we're going to investigate that, the first thing we can do is we can look back, right? Like our view of walking up that hill, we can look back and we can say, what can we learn from the process of evolution that has already happened and has got us to where we are today? And I think it's worth reflecting on the fact that all of us here in this room or listening online are essentially a statistical anomaly, right? We are a tiny, tiny proportion of all those humans that have gone before us and have died. We are the survivors because we are descendants of people who had really fit genes. And the vast majority of all the other people back in those generations died and did not produce offspring. So we're the kind of lucky few. So going forwards, that will be the same case, right? There will be bottlenecks on selection going forwards. So if that happens regularly, we should be able to see that kind of imprint, if you like, in modern populations. And the good news is we can. So here is an analysis done by David Reich's lab a few years ago. And their principle was quite straightforward. We get a lot of infections, right? Everyone does, even today. Historically, many of those infections will kill people, right? Today we survive a lot of infections, but historically, before antibiotics, before vaccination, they were the major killer, as you just saw. Therefore, you would expect that genes that confer an advantage against infection will be evolving faster than other genes. For example, genes that control, the way you metabolize fat or your eye color, they're not really going to make a huge difference to your survival advantage. But a gene that helps you survive some ghastly viral infection is a really big benefit when it comes to evolution. So you should expect that genes to do with immunity are evolving faster than other genes, and they are. So this sort of diagram, I'll show you a couple of these, so let me explain it. This is essentially a plot of genetic variation for every single gene in the human genome, and we have about 20,000, right? And so it's arranged by chromosomes. So imagine all your 20,000 genes in a long line here going from 1 to 20,000. And then what we say is we sequence a whole bunch of people and we say how different is each of those genes. And if all the genes are very similar, they fall down here in the noise. That's just background fuzz. And if you suddenly see a lot of diversity, more than you would expect by chance, you infer that that gene is evolving fast. And what you can do to make this more sensitive is you can sequence people who died a long time ago. And so in this particular study, they sequenced people who died up to 3,000 years ago, so mummies primarily. And what you say is, you look at those genes and say 3,000 years ago to now, how much change has happened in the genome? And what you see is for most genes, very little has happened. They've just been bubbling along the bottom. But there are these occasional ones that shoot up the y-axis, and they're evolving far faster than you would expect by chance. And in accordance with our prediction, almost all of these have a role in your immune system. So for example, I've just picked out 2 here. So this is a group of genes here, TLR 1 to 6, and those are involved in sensing the presence of bacteria and viruses and alerting the immune system. And this one over here, MHC, we'll talk about it a bit later, that is one that's to do with revealing the presence of intracellular viruses. So if you have a virus that's hiding, these genes help reveal it to the immune system. And you can see that they are evolving much faster than by chance. So the immune system, our first hypothesis is true. The immune system is evolving faster than many of the other functions in your body. What we don't know from this study, of course, is why. All we know is that somehow these genes have conferred an advantage to us. That's why we're here today. And people thousands of years ago died from something, but we don't know what the something was. But you can do this in a more sensitive way if you know a particular signal of a particular infection that you want to look for. And that has been done in a really elegant way quite close to here. And it's been done on perhaps one of the most significant infections of human history, which is the one that's carried by these guys. Actually, these are the innocent bystanders. This is the rat, of course. And rats were accused of carrying the bubonic plague or Black Death. Of course, it's not actually their fault at all. It's the fleas that they have on them that carry the plague. And in fact, it's not even the flea's fault. People underestimate fleas. The flea also gets sick when it has bubonic plague. You've got to feel sorry for the fleas. And in fact, they get so sick they can't swallow. So one of the reasons that you get bubonic plague, hopefully you don't, is that the fleas, when they bite you, they actually vomit back up the bacteria because they can't swallow properly. So feel sorry for the fleas at the same time. However, those of you who've studied your history will know that the Black Death or the bubonic plague has swept across the world in repeated cycles over hundreds of years. And the one that we particularly think about in Europe is the Black Death of the medieval period, which occurred in multiple waves, but was responsible for, given the circumstances we're here today, the Plague of London, for example. So, the Black Death has swept through. We have really good historical records of it being quite a dramatic selective force on humanity. So, for example, the first case we know of with reasonable accuracy in history is the so-called Plague of Justinian back in the Mediterranean. And that's estimated, based on records from Roman and Greek writers, to have killed about 1 in 4 people across the Mediterranean basin. And the one that we're talking about now, the Black Death, which occurred in multiple waves, but particularly a really strong wave in about 1348 to 1350, swept through Europe and had an estimated mortality of between 30 to 60%, and in some parts of Europe, essentially extinguished entire villages and towns. This is a huge, huge mortality rate. And it means that it's a huge, huge selective force. Because if you've got a 50% chance of dying, if you happen to have a gene that confers even the tiniest advantage, it will be very, very strongly selected, right? Because you've got a 50% chance of dying, but if it turns out to be 49%, that's a whole lot better. So we would expect that when the Black Death came through, bearing in mind there were no vaccines and no antibiotics, the driver of whether or not you survived if you were exposed to the Black Death were your immune genes. So you would expect that this might have left a strong evolutionary signal. The question is, can you test it? And it turns out you can test it in an amazingly elegant way just round the corner from here. So this is a study from Luis Barreiro, based on a mass grave just here in London, actually, which was dug during a particularly profound wave of the Black Death in 1348. It was dug on orders of the king because Londoners were dying left, right, and centre. And he said, "Oh, this is too bad. We can't have people going to graveyards. Forget all that. None of this church business. We're going to dig a really, really big pit, and we're going to put all the bodies in there, and we're going to cover it in lime because in those days they thought that was quite a sterilising thing, and then we're going to fill it back in again." So they put all the plague victims in the thing. They covered it in lime, and then, as many a king and prime minister subsequently has done, they've gone, "Hey, that was quite a good idea. We've spent all this money digging this pit. Don't fill it in just yet. We'll keep that because more people will die and we can just keep topping it up." And of course, the Black Death had passed through, but people die of other things. And so in the subsequent years, Londoners continued to die, but not of the Black Death. And they were added to this pit. So in that pit, you have a layer of people who largely died of the Black Death, and then a layer on top of people who died a couple of years later of something else. And that is a fabulous resource, because what you can do now is you can sequence the genes of those two layers, and you can say the ones who died of the Black Death, they would have had genes on average that made them susceptible to the Black Death. The ones that died a couple of years later presumably had genes that were beneficial in surviving the Black Death, but not whatever killed them in the end. And you can compare those two populations. And that's what's been done here. And it's a fabulous study because what it reveals are a number of genes that are very, very strongly evolving between these two different populations. So they have a big advantage, the ones in yellow here. And what you can now do, of course, with modern methods is you can take these different gene variants and you can test them in a lab. And sure enough, it turns out that many of these gene variants do indeed help protect you slightly against the Black Death. And that's a terrific advantage. That is part of the reason that many of us are here today, because we are the descendants of these survivors, if you like, who had these good genes. It does have a slight cost, and we're going to come back to that later, because many immune genes have a sort of yin and yang. They have a benefit but a disadvantage. And it turns out, for example, that a couple of ones that are highlighted in yellow here give you a benefit in terms of fighting off the bacteria that causes the Black Death, but they also make you much more predisposed to things like rheumatoid arthritis. So for those of you listening in the audience who are complaining about your arthritic joints, I feel very sorry for you, but also you do have to give a little nudge of positivity, because it's probably the fact you've got rheumatoid that means you're here at all, because the same gene helped you survive this several hundred years ago. So this idea of trade-offs in evolution is something we'll come back to. So this is all very historical, and you're all sitting here thinking, I want my money back. It was a free ticket, but I want my money back anyway, because he said he was going to talk about the future, and now he's talking about, for goodness sakes, the Black Death in 1348. What is this lecture about? And the reason I've told you all that is because it sets into context, I think, when we're thinking about evolution going forwards. And of course, the thing that is high in everybody's mind still now is the very significant infectious disease outbreak that we've all just lived through, the COVID-19 pandemic. And so COVID-19, like all major infections, has been a selective pressure because some people have died and some people have survived. And I think in no way to diminish the huge personal tragedy and the global impact of COVID-19, it is important to set it in context that overall, we estimate that COVID-19 has killed about 15 million people worldwide. So that's about 0.2% of the global population, which is a tragic number, but is orders of magnitude less than something like the Black Death. which means that the evolutionary signal of COVID-19 will be much, much weaker. And I suspect in hundreds of years' time, it might be essentially undetectable. I'd be quite impressed if people can see any sign of evolution as a result of COVID-19. Who knows, though? But the good news is we can do an experiment using the kind of COVID-19 example, which doesn't require us to dig up lots of dead people from graves, happily. And instead, we can look at how genetic variation might have influenced survival during the pandemic itself. And the way that's been done is this very nice study here, where people looked at COVID-19 patients in intensive care. And so the theory was, many of us in this room, I'm sure, will have had COVID-19 and hopefully probably felt a bit rough, but didn't even have to go to hospital and certainly didn't end up in intensive care. But we will also know people who might have been incredibly fit and suddenly ended up in intensive care. And so the question is, what is different? Is there some genetic variation between people who had mild COVID and people who had very serious life-threatening COVID? And so you can do what's called a genome-wide association study. You can look for all the genetic variants in a whole bunch of people who got COVID but had mild symptoms, and a whole bunch of people who went into intensive care. And you can say, are there any consistent trends? Are there some gene variants that predict you're more likely to end up in intensive care with COVID? And of course, the answer is yes, otherwise I wouldn't be showing you the slide. So on this very similar layout, again, all the chromosomes from 1 to 22, all the genes as dots, and you can see at the top a few genes that are above that red statistical line that predispose people to having severe COVID. And unsurprisingly, most of them have some role to play in the immune system. So for example, OAS1 over here is a viral response gene. And this has been, so if you kind of play forwards the tape of evolution, if we hadn't have had a vaccine and if we hadn't have had good medical care, ultimately COVID-19 would have killed a huge number of people, and it would probably have reduced the frequency of these genes in the human population. And just like the Black Death, we would see that signal in terms of survivors today. Fortunately, we didn't get there because we have much better medical care, and so many, many people with these gene variants survived COVID-19, but you can nonetheless see that kind of signal of susceptibility. And one of the really neat things about this, I think, is that this opens the door to some really clever medicine. And in this particular example, they used this study to help guide patient treatment, because it turns out that one of the genes they identified up here, TIC2, is a gene that, again, predisposes to severe COVID. And it appears to do that by being slightly overactive all the time. So it's kind of driving too much of a response. Fortunately, this is a gene for which people have already developed an inhibitor for, because it's implicated also in multiple sclerosis. And so there was a drug that had been developed for treating multiple sclerosis that works through targeting this gene. And so they were able to use that, it looks like this, you can use that drug in the clinic to treat people with severe COVID-19, and it worked. And so this, I think, is a great example of where the future of immunity might go. This idea that rather than taking all patients and treating their symptoms, you work out their kind of underlying physiology, their underlying genetic variation. You do some gene sequencing, and then you target a specific drug to the specific defect or change they have in their metabolism to help them get better faster. And I think that's a really kind of exciting idea that we'll return to in a minute. So here's my first prediction. I said I could do some wild predictions, and well, maybe you can come back in 50 years and tell me if I'm right or not, but I suspect I won't be here. So here's my prediction. My first prediction is that I think, at least in developed economies where we have more healthcare money available, in the pretty foreseeable future, your visit to the GP, or indeed to the emergency room, to A&E, will look more like this than it does today. Because I think there is quite a lot of merit, if you would go to your GP, for example, with a chest infection, as the price of DNA sequencing falls and falls and falls, and the speed of sequencing gets faster and faster, I don't think it will be very long before you go to your GP, and before you even see anybody, if you see anybody, they take a drop of blood, they do your DNA sequence, they check for the variants that might predispose you to influenza or adenovirus or whatever it is. And they work out, are there any drugs to match these particular variants? And by the time you go into the GP's office, oh, bad luck, Mr. Smith. No, not you, Richard. Bad luck, Mr. Smith. It turns out you have this interesting gene variant here. The good news is I've got 3 drugs which used in combination will help treat your infection in a very customized way. And I think it has huge potential and quite a lot of excitement. So there's my first prediction for 10, and let's see if I'm completely wrong in 20 years' time. Okay, so far we have talked about all the great things the immune system does for you, largely speaking, right? It fights off bubonic plague and COVID and all these other things. But I suspect all of us in the room pretty much have also had experience of the immune system not behaving itself, and in particular this one here. You show this nice scenery, and I suspect many of you in the room go, oh, that looks very nice, I'll go there on holiday. And I bet quite a few of you are looking at that and thinking, this is my idea of a nightmare, because I will be sneezing and spluttering my way through this entire field. Am I right? It's good hay fever season, this one, right? A few nods in the audience. Yes, it's a very good hay fever season. Hay fever is probably one of the most widespread examples of your immune system getting it wrong, right? Because what your immune system is doing is it's responding to a totally harmless thing, like pollen, as if it was an infectious threat. Hay fever is, for most people, at the kind of mild end of, it's irritating, but it's not life-threatening. But of course, there are lots and lots of autoimmune diseases where your immune system goes wrong, which are much more severe and much more life-threatening, ranging from things like multiple sclerosis through to very severe food allergies, for example, like anaphylaxis. So, might we be able to harness our kind of new knowledge of the immune system to improve our treatment of these diseases too? So not infections, but to harness our ability to control the immune system and prevent it from damaging us. To some extent, we are already starting to do that. So, for instance, the idea of tolerizing therapies is just kind of coming to the fore. So, for people with particularly severe food allergies, under clinical guidance, you can now, in some cases, be exposed to absolutely tiny quantities of the thing that you respond to, so peanut, for instance, in a way that tries to train the immune system to dampen down its response. That there, and I do want to really stress for those, especially for those listening at home, this is not something you should try at home, right? If you have a severe food allergy, please don't go and try a peanut just in case. That's a very bad idea. But under clinical guidance, this is a therapy that for some people is working quite well in retraining their immune system, but not for all. But the question is, in the future, might we be able to do this better? And might we, for instance, be able to take the same approach that we've taken for things like COVID-19 and work out what it is in your genome that makes one person much more susceptible to an allergy than another? And that is just starting to happen. So here is a similar GWAS study. I said I'd show you a lot of these plots, and I am. This one is for an autoimmune disease that is very, very common for many of us, which is asthma. Right? And asthma ranges from relatively mild, well-controlled to very severe, life-threatening autoimmunity. In asthma, what is happening is your immune system is responding typically to a harmless threat, like a dust mite or a pollen, and triggering this profound, very rapid immune response that can sometimes kill you, essentially. So the question is, can we identify immune factors that drive this runaway effect, and can we do anything about them? And what you can see here, so a similar approach where we're looking for genes that are different in asthma sufferers versus those who don't have asthma. And the first and slightly disappointing thing, although quite predictable, is that there are an awful lot. So you can see lots and lots of these genes above the line here. And this is a classical, what we'd call polygenic disease, right? This is not a disease like cystic fibrosis, where one gene is wrong and you can fix it. This is a condition where many, many genes are all interacting together and causing the syndrome together. So the bad news for that means that there's no kind of magic bullet. You can't just take out this one and solve someone's asthma. But what you can do is look for things that are druggable, if you like, genes here that might be approachable as a target and may not cure your asthma, but might dampen it down. And for some people, of course, that could be a game changer. If you could change your asthma from being life-threatening to mildly debilitating, that would be a big, big step forwards. And so the first studies to do that have just been reported, actually. And one of the genes they've looked at is this one here. So this is a protein called CD52. It's present on your white blood cells. And it seems to be in people with asthma that this is kind of hyperreactive. It triggers your white blood cell to respond too fast. And there has been a drug developed to target this protein, again for other purposes. And at least in mouse models, it turns out that if you use this drug, it will dampen down that CD52 response and reduce the symptoms of asthma, at least in mice. And I appreciate we're not all mice, so don't get too excited yet. I think this is very promising. It is not the solution, not least because CD52 does lots of quite important things in your immune system. You don't really want to dampen it down the whole time. So I think, but as a proof of concept, I think it's quite exciting. And maybe 5, 10, 20 years from now, we'll have decoded more of these and worked out a much more kind of tailor-made way to reduce people's asthma susceptibility in a way that doesn't impair your immune system more broadly. So I think this kind of genome-wide approach, this strategy of kind of bespoke personalized medicine is quite a promising one for the future. So that's all sort of, broadly speaking, evolution. It's about natural variation in genes. It's about naturally harnessing the biology that's out there. But of course, the really exciting thing for people in the kind of medical world is not just what nature is doing, but what we can do to improve it. And the question, I guess, is, to what extent can we engineer our immune systems to get better? And as I've said at the beginning of the lecture, up until now, so for millions of years of evolutionary time, the only ways that genes have changed in our genome is through natural selection, through either death or reproduction, right? And that's all that's happened for millions of years, until about, 20, 30 years ago, when we started to harness the power of recombinant DNA technology and gene engineering. The ability to change in a specific way DNA sequences in all living organisms, including ourselves, if we want. And in particular, so we've had genome engineering for pretty much most of my life in some form or other, although admittedly it's become unrecognizably better over that period. All of which has been a very useful tool. But the real game changer, I think, particularly for medicine, is this particular type of genome engineering, CRISPR-Cas9, which, as you may know, was discovered just over a decade ago, and its discoverers were awarded the Nobel Prize. And the really key thing about CRISPR-based gene editing is that now, instead of taking whole chunks of DNA and cutting and pasting them in or out, you can edit individual letters of your DNA code with huge, huge precision. And that's really important, because if we think about all those diseases and those risk factors I've just been showing you, it's not that if you've got asthma and I haven't, it's not that you've got a CD52 and I don't have it. We both have CD52. It's just that your variant is tiny, tiny bit different to my variant. And so what you want to do to have an impact in immunity is to be able to edit that sequence in a very, very accurate and tiny way. And that is what this tool allows you to do, because it allows you to edit individual letters of the DNA code. And that has been largely a lab-based tool for most of its 10 years of discovery until recently. And you may know that the UK became the first country in the world to authorize this as a medicine for treating humans. And in particular, this is for 2 genetically inherited blood disorders where we know exactly the mutation, the DNA mutation that has gone wrong in those patients. And we can correct it using CRISPR-Cas9. So the way this works is you put the machinery, the molecular machinery into the patient. It will edit back their DNA. It will correct the error. And it will restore the function of the normal gene. And it will essentially be a lifetime cure for those people. And there's a really important point about this that we'll come back to in a minute, which is that this happens at the level of the tissue. So it's happening in the bone marrow of the patient, which means that you as a patient are cured because your bone marrow is now corrected and it's producing fully functional red blood cells. But it hasn't changed your eggs or your sperm, so your children will still inherit the faulty gene. And we'll come back to that in a second. So the question now is, so we've started to do that for these diseases, these blood disorders. There are a whole host of things where I think you could apply this to immediate effect, if we had the technology and the resource to do so, with real advantage. So today, before you have a baby, if you choose to have a baby, particularly if you're slightly older parents, you will be screened probably for a whole variety of possible genetic disorders. There's something like 50 disorders that are tested for now, many of which are single gene disorders, so things like cystic fibrosis, for instance, and many of which are amenable to this kind of technology. So I think it probably won't be very long before we start to offer these kind of gene editing techniques to correct, for want of a better word, gene sequences for a whole range of disorders. But you could also imagine doing this permanently, right? So if you're someone with beta thalassemia, that blood disorder, you want to be cured, but you also don't want your children to have this disease. And so there is, I think, a valid ethical argument to say, well, what about editing that gene in my offspring too, in my egg or my sperm cells, so that my children are born free of the disease and wouldn't ever have to suffer it, let alone go through this treatment. And that raises a really interesting ethical dilemma, I think, because for that individual, it's overwhelmingly ethical, right? You don't want to have a child who's sick, most of us want to have a healthy child. For society as a whole, you could argue it's also in our interest. We want a healthy society, and very practically speaking, we don't want the cost of having to look after people who are sick. But you would be changing permanently the evolutionary process of genes in that offspring. It might be only one child, or it might be 10,000, but you will be making a change that has not happened by natural evolution. And for the first time, you would be sort of shaping the future of human genetic diversity based on those kind of edits. And that raises all sorts of really interesting and tricky challenges, I think. So how might you apply that, though, if you wanted to kind of reshape immunity? And what you could do, for instance, is you could look for genes from those kind of studies we've just identified that you might want to edit to a more functional, to a better variant. And I said, for example, in the case of asthma, where you have lots and lots of genes, at the moment, our ability to edit genes is pretty slow. So we're basically limited to doing one or two genes. But it won't be very long before we start to be able to do this very efficiently. And you might be able to take all of those 150 genes or whatever it was associated with asthma and systematically edit every single one to a better variant and cure, in inverted commas, someone's asthma. So here's a good example. So here's one of the proteins that is involved in these autoimmune disorders like asthma. Okay, we know this is the structure of the protein, and we know there's a particular mutation that is associated with asthma. So if you have a particular change in this protein, you're more likely to have asthma. So you might come in as a scientist like me and say, well, here's an easy solution. I'll use CRISPR, I'll correct that mutation, and we will help solve your asthma. And that would be a great idea. Except that if you look a bit deeper, here is the gene concerned, right? Mapped the DNA across here. And in blue are all the identified mutations that we can see out there in the normal, healthy human population. And there are loads. So I've identified one of these that I could correct, and I might be able to correct that, and that would be great. Maybe that will help with asthma. But it turns out that all these other ones do all sorts of other things. In fact, variations in this gene are associated with cardiovascular disease, TB susceptibility, et cetera, et cetera. And sometimes the same mutation goes in two different ways. And so this is really the core of the problem. If you are to make a edit in an individual, let alone a permanent edit that would change their offspring's inheritance too, you need to be absolutely sure that the edit you're making is going to cure one problem but not cause another one. And at the minute, we don't have enough information to do that, because you could naively correct someone's asthma but then give them a thyroid disorder, which is definitely not ideal. And so before you're going to do these kind of massive steps that are essentially irreversible, like editing someone's DNA, you really need to be pretty confident that you've got the right candidate. So, those are the kind of edits for things that we come across every day. But the thing that I said at the beginning of this lecture is, what about the kind of most negative prospects? So what about, we're all sitting here happily, hopefully enjoying a pleasant summer's evening in London, not worrying too much about the end of the world, but it might be just around the corner. And in particular, of course, being someone who works a lot on infectious diseases, I think a lot about all the horrible infections out there that might end the world. And you'll be delighted to know there are loads of them. There are many, many ways in which civilization may end, some of which are really quite boring. So this is antimicrobial resistance. So here we're looking at E. coli and Klebsiella. So these are two common bacterial infections. And this is a survey of individual patient isolates looking for resistance to antibiotics. And it's really depressing because what it shows, as you can see on the graphs, is that actually if you go out there and you survey lots of people who are sick with bacterial infections, lots and lots of those isolates are already resistant to antibiotics we would otherwise use clinically. And I'm sure everyone in this room is aware of this kind of growing massive threat of having untreatable bacteria. We're not very far away from moments when you will go to the doctor with a really mundane thing like a urinary tract infection, and they might say, I'm sorry, I've got nothing to treat you with. Good luck. Because evolution has shaped this huge resistance. And some of the threats from AMR, for antimicrobial resistance, are they're almost laughably daft. So this is one that worries me quite a lot in my day job. This is Candida auris. It's a fungal infection. And this is actually not a result, we don't think, of human selection. This is an organism that appears to be intrinsically resistant to most of the antifungal drugs we use. And this is relatively recently discovered, very bizarrely, in the ear of a Japanese lady, hence auris, where it caused this very, very difficult to treat ear infection. And originally, it was only about 10 years ago or so, originally it was thought of as being a bit of a kind of random infection, let's not worry about it. Turns out we took our eye off the ball and it swept quite rapidly around the world. And you might be familiar with the fact that several hospital wards, including in the UK, have had to be closed in recent years for incredibly deep cleaning because they've got this organism in the ward and we can't treat it. So a really depressing possibility is we might be driven to extinction by a really boring fungal ear infection if we're not careful. Or, of course, we might be driven to extinction by something which is much more exciting if you're a Hollywood film director, some emerging horrible pandemic. And this is not just obviously the stuff of science fiction. This is the basis of COVID-19, which jumped from an animal reservoir to a human reservoir. This study, which sadly has been discontinued, but was driven by the US, where they were out surveying particularly rainforests for potential new human viral infections. Over the 10 years that it ran, identified almost 1,000 new previously unidentified viral threats to humanity, which is quite a big number. And of course, this potential for something to jump into humans and cause disaster is rising all the time because we're doing things like this. We're cutting big roads through the middle of rainforests, encountering organisms that may harbour diseases we haven't seen before. And we also do things which are much more mundane, but are also a major threat in terms of infection, like intensive agriculture, where you have sometimes millions of animals in a very tightly confined space. It drives very rapid pathogen evolution. And of course, we do the same things with ourselves. He says to a relatively crowded room, we squeeze ourselves together and we cough and splutter over each other on the London Underground or wherever, driving very high rates of pathogen spread. So the next major pandemic might be just around the corner. It might emerge, and that would be quite bad, need to say. But it would, for evolutionary biologists, be at least a very interesting experiment in how your immune system might help you resist things. However, to avoid that experiment, what might we be able to do to head it off? And one of the things I think we could do is we could get a lot better at predicting things. And this was a question I had one of my earlier lectures. Thank you for if you're back in the audience, whoever asked this question, which is, can we use those kind of techniques to understand more about predicting and averting future threats? So this is currently, I think it's still our number one threat on the UK biological risk register. This is influenza, flu. And still, one of the biggest threats we face is the idea that a very virulent form of flu will jump into the human population and cause huge mortality. Bearing in mind that, for example, the 1918 flu, which swept after the First World War, was about 10 times as lethal as COVID-19, right? So the big risk with flu is that flu circulates typically, so we have seasonal flu in humans, obviously, the one that you get that makes you feel grotty but doesn't usually kill you. There is flu circulating in bird populations, which, if it got into a human, is much, much more severe, but generally stays in birds. And so the big risk we have is that it will jump from a bird to a human. And we know exactly how that happens, because the reason that bird flu stays in birds and human flu stays in humans is that we have a different sugar on the surface of our lung cells, and it's the sugar that lets the virus in. So if you're a flu virus, you need to bind to this sugar here. Sorry for the horrible diagrams. You need to bind to this sugar here. And so if you're a bird flu, you attach to this one here. But if you get into a human, we have this ever so slightly different sugar up here, and you can't bind to it. So you stay in a bird, and vice versa. So the big risk to us as humans is that a bird flu variant will evolve the ability to bind either both of these or the human ones, and it will jump into humans. And that's the reason we do all this surveillance to try and look for new flu variants in poultry, for example. But it's also the soft spot, because we can look, for example, excuse me, for a moment, we can look in bird populations at the minute, and there is, as you will be aware, a circulating pandemic of avian influenza in bird populations around the world. And we can say, okay, what does the virus look like? And then we can predict what might it turn into if it was going to jump into a human. And so you could, theoretically, use that power of DNA sequencing and predictive modeling to design a vaccine for a virus that doesn't exist yet, but it's ready, right? So we could design a vaccine now that is ready for when evolution does what we think it might do and jump into a human. And this, I think, is a really exciting prospect for the future of immunity. Then we can go one step forward because, as you will all know from COVID-19, we have this fantastic new technology of mRNA vaccines and viral vector vaccines like the Oxford vaccine, where you are harnessing the power of other viruses, harmless viruses, to deliver vaccines very effectively. And so now you have all these tools together, you can combine them. So what you can do is you can take massive amounts of DNA information, because DNA sequencing is really, really cheap, and you can look, for example, at all the possible influenza variants that are out in nature at the moment. Then you could use some really fancy modeling by AI, for instance, to work out all the possible trajectories of evolution and say, okay, which of these changes might be combinable in a way that's likely to happen? And you could predict, therefore, what the most likely next pandemic virus is, and you could create a vaccine, or possibly even a vaccine library, now that DNA synthesis is quite cheap. So you could, if you wanted to, make thousands and thousands of different variants of vaccine ready to go, and then the minute that someone starts coughing in a horrible way at an airport, you sequence them and you launch the vaccine that you've had prepared already. And I think this is really exciting. It is not cheap, and that is the problem, but pandemics are also not cheap. So I think there's a real question for international governments here about how much money you spend on this and how we might predict it. And of course, one of the challenges is what is a major threat to, for example, us in the UK is quite different to what might be a major threat in somewhere like Ghana. And so you have this kind of complex diplomacy around working it out. But that's my second or possibly third prediction of the lecture. The other thing that we could think about, and this is much more challenging, but someone asked me this question last time, so I thought I'd cover it, is what about if we go one step further? What about if we make these viral vaccines, which we already do? At the moment, those viral vaccines are deliberately engineered to not replicate, right? If you got the Oxford vaccine, like I did, that virus goes into your arm, it vaccinates you, and then it dies out within a few days, deliberately as a safety measure. But you could perfectly reasonably engineer that virus not to die out, but to spread. Because that would have a huge advantage, because then I could immunize one of you in this room against influenza, and I could go away and have a cup of tea, and by the time I've come back, you've coughed and spread it over everybody else, and they've all got immunized too, and for the price of one needle, I've vaccinated however many of you there are in the room tonight. And that, I think, is potentially very appealing, but of course raises massive questions. The biggest question of all is about consent. Because if I vaccinate this lady in the front row here, she has to give consent, but if she then coughs over the lady next to her, bad luck, you've been vaccinated whether you liked it or not. So I think there are huge barriers to this in humanity, and I suspect it's very unlikely to happen unless, of course, things get out of hand. If you were in a pandemic situation where you were unable to control it, I could imagine some governments, particularly more authoritarian governments, saying, bad luck, no choice, here we go. And in particular, we might start doing that for things like livestock. I think there's a stronger argument at the minute to do self-spreading vaccines in things like cattle or poultry, possibly. And finally, then, we might be able to tackle all these things. We might be able to head off pandemics. What about some of the stuff that's not infection related? How can we harness our immune systems to tackle some of that? And we talked a little bit about this in my vaccination lecture a few months ago, where we are just starting to see the technology that we use for fighting infections come into non-infectious diseases. And the big one, of course, that I'm sure you've all heard of is the idea of immunotherapy for cancer treatment. This is a skin cancer here, so you can see a healthy skin biopsy and clearly a cancerous skin biopsy here. We talked a bit about the fact that it's a real challenge to do this because by definition, cancer cells are your cells. Right? So if I'm going to vaccinate you against a cancer, I somehow have to get you to mount an immune response against your own cells, but not in a way that damages all the healthy cells around the outside, just the tumor. So what you're looking for in a cancer vaccine is what's called a neoantigen, something that the cancer cell produces that your healthy cells don't. So for example, a mutated protein. But that is happening, and as you'll be aware, actually, this is showing huge promise. So for some cancers, cancer vaccine therapy is actually really, really powerful, particularly ones that are difficult to operate on through other measures. And I think over the next 10 or 20 years, we'll see increasing rollout of cancer vaccines as treatment. We may start to see increasing rollout of cancer vaccines as prevention too. Those of you, if you're listening, who are relatively young, might already have had, for example, the papillomavirus vaccine, which is designed to stop you getting papillomavirus, not because the virus itself is particularly problematic, but because it is a big cause of cervical cancer in adulthood. And so you're preventing a cancer by being vaccinated in the first place. But the really big goal, I think, and the one that I am particularly excited about, although we're still a very long way off, is can we harness this sort of technology also to tackle some of the biggest scourges of old age illness, and in particular, these. This is Alzheimer's disease brain biopsies, and you can see these classical plaques, the purple and the brown here. As you know, when Alzheimer's kicks in, what is happening is your brain is accruing these kind of lumps, if you like, these lumps of non-degradable tissue. Bit by bit, the cells start to die and you start to lose mental faculties. The rate of senile dementia is generally rising right across the world, particularly in developed countries. However, there is some promising, very, very early data that you might be able to encourage the immune system to identify these tangles and clear them at an early stage. And I think, to me, that's one of the most exciting prospects. Maybe in years to come, you'll be getting vaccines that prevent you, when you're 10 or 20, that prime your immune system to prevent any of these things accumulating long before you have any symptoms of memory loss, and therefore we live a long and happy life, Alzheimer's free and everything else free. And so my dream ticket to finish on is that in years to come, we'll be rocking up to our GP, we'll be getting stabbed in the arm like this is the South African president. Doesn't look very happy about getting his COVID vaccine, but it was supposed to be advertising. And it will protect us against flu and COVID and cancer and Alzheimer's and everything else. And we will all be coming to Gresham lectures when we're 305 and reveling in it. Who knows? Thank you very much indeed.